they aid in attachment. They protect the cell, so that's not written on here, but it'll be on each one of those. So you might as well add that to protection. I mean, that makes sense that you have anything outside of the cell is going to have some protection. So when it says aid in attachment to solid surfaces, that means it could be something that is allowing it to attach to, whatever substrate it's on. It could also allow it to attach to other bacteria. So when it says to solid surfaces, that's really only one part of it. It just basically, they aid in attachment, period. Attaching to each other, attaching to any kind of substrate they might be on, whether it's a solid surface or another bacterium. It says for the example biofilms, and I've mentioned like last time biofilms and kind of said, oh, that's right, we haven't really talked about biofilms yet. And biofilms really are covered later on in the textbook, and they're mentioned maybe at another part in this chapter too. So I do want to just make sure that we kind of briefly introduce it so that when this term comes up, you know what I'm talking about, but it will be written into the notes a little bit later as well. A biofilm basically is a collection of different bacterial cells that they stick together and then they start kind of functioning together. We used to call biofilms basically like clumps of organic material with bacteria. So like if you looked at, say, something like water, and you had sort of clumps of organic material that was in that water, whether it was just like algae or other types of nutrients or something like that, then it would allow different bacteria to kind of connect to it, and then they would start growing and they would be sort of protected in that clump. But as the years went on, they really realized that there is something really significant about biofilms per se, that you have different bacteria that, like, you know how bacteria grow in a colony? These are different bacteria, different species of bacteria that basically are growing in something similar to a colony. Then together they influence each other and they start to actually function kind of as one unit. This is something that actually probably the best example of a biofilm that you would all be familiar with is plaque that grows on teeth. Basically what happens is you have different bacteria, and then together they start to secrete other substances that protect the colony, so to speak. And they will send out organisms to kind of break off from that colony, and they continue to grow. That's why something like plaque on teeth and other biofilms are especially hard to combat, because the substances that they secrete really protect that colony itself. So it's kind of, I mean, it's sort of a little bit misleading that that's the example that they mention here as far as like the glycocalyx in general. But any time that bacteria are attaching to something and starting to grow, whether it's in a biofilm or it's in a colony, so to speak, the organisms that are going to be in the center of the colony are going to be a lot more protected than organisms that are on the outer part of the colony, right? So if you can imagine that if you had, okay, well, here's an example in your kitchen, right? You got your kitchen counter, you're cooking, you get little pieces of organic material on the counter, right? Then maybe there's some bacteria on there, and those bacteria might be starting to grow because maybe you haven't cleaned off the kitchen counter or something like that. Maybe it's not you. Maybe it's like somebody's apartment or the dorms at a college or something like that, and they don't clean it up, so it like sits there for a while. Then if somebody just wipes it up, aren't you just going to wipe off kind of the stuff that's at the top, but you might not necessarily like get to everything on there? So you could imagine in your own kitchen, if you were working with something that was really messy, there could be organisms that are underneath some of that organic material that are going to be protected during the process of the time that you're early on, I mentioned something about how many people have ever read on the container of a cleaning solution, you know, for your house, like how you're supposed to use it to disinfect. And I said that if you read on there, and maybe some of you now since have done that, first you have to clean the area with that cleaning solution. Then you have to spray it again and give it a contact time. That's so that maybe that solution can penetrate to get into the organisms that are underneath the top organisms. Does that make sense? And if you picture your colonies, you can picture how organisms would grow kind of in a real world situation. So on top of just that, you have the glycocalyx. The glycocalyx is going to add so much protection to the bacterial cell that you could be in a situation where if I was the inside of the cell, all the way to the end of this room is the glycocalyx. Especially organisms that are living kind of in the real world situation where they're having to survive in something less perfect than a culture tube, where all the nutrients are there, all the moisture is there, and the temperature is just right. Organisms that are actually living in areas outside of that kind of perfect environment might be utilizing energy for other things like an extensive glycocalyx. That's why they've been able to find things like Pseudomonas aeruginosa surviving for 14 years in disinfectant. Can you imagine that? 14 years, no nutrients, living in disinfectant, living in iodine. Okay, the only way that happens is with a glycocalyx. You have the glycocalyx that's so large that the organism that's inside there is completely protected from those things on the outside. And if you're really talking about those kinds of organisms that are, say, living in a hospital environment, they are going to produce a very extensive glycocalyx and be able to survive in, you know, much more difficult types of environments. So they're much harder to kill off. And then if you add the fact that they could be in a biofilm or just simply a colony, then they're even that much more protected. That's why, like, keeping everything clean and free of organic material in itself is something that's going to reduce growth because you don't have the protection. On top of that, we'll find out that a lot of disinfectants are really inhibited by extraneous organic materials, because those organic materials that are just kind of sitting around will actually combine with the molecules in some of the disinfectants and make them inactive. With just this kind of information, the practical aspect of it, I think you can see, right? This is just something that affects you in your everyday life, but for sure will affect you in an environment like a hospital environment. So everything that you read about, everything that you know about the organism doesn't necessarily take into consideration what that organism is like in a real world environment. So about 10 years ago, maybe a little longer than that, they realized, the EPA realized that so much of the research that had been done on disinfectants and antiseptics was extremely flawed. And can you imagine why it might be so flawed? Because how do you really test for that kind of condition? If you're in a lab and you're doing these tests, aren't you going to have to grow the organisms in an ideal environment? Because otherwise, how do you culture those organisms? And then, are those really the same organisms that you would find if you were in actually the real environment? Does that make sense what I'm talking about? These are the kinds of things, like, remember the first exam where I asked about the mycoses and what different factors would make us more concerned about them? When we're learning about the bacterial cell as we go, I'm trying to give you practical information of knowing about the cell itself helps you understand the difficulty that we have in trying to control the growth of organisms. Because you can't really control for that. And we haven't really, like maybe I threw it out there, but bacteria have the ability to undergo hypermutation, which obviously isn't really related so much to the glycocalyx, but it could be related to any factor in the bacterial cell. You know how our cells will undergo like some kind of spontaneous mutation, like maybe every million cell divisions? Remember like learning that in 107, something like that? Bacteria will undergo the same kind of spontaneous mutations, but when they're in a really adverse environment, they'll start undergoing mutations a whole lot more. And then finally one of them will end up maybe developing a mutation that allows it to survive, and then it'll start then undergoing binary fission. How can you control for things like that? You really can't. That's why you have to be knowledgeable about this, because you can't just rely on the fact that everything is known about everything. You always have to just think, it's much better not to get the contamination. It's much better to really thoroughly understand aseptic techniques so that you don't end up in a situation where there's some kind of infection that you get, or your family member gets, or somebody that you're working with gets. Because after that, it's a lot harder when you have all these different factors that affect how organisms can survive. It just kind of puts you really in a more concerning situation. So better to understand all of this. So I could, as we're going through and I'm talking about things like this, it's so that you can understand how this really relates to every day. So you guys, you get it, right? So we'll go through each one of these, the capsule, the slime layer, and the S layer. But when it comes to their function, mostly their function is aiding and attachment. And aiding and attachment not only helps them attach to things, but also gives them protection. Just the fact that they're attached to themselves and other bacteria and other substrates, but also the fact that they're all coated with another layer also is giving them protection. So first, the capsules. So the capsule you see here usually composed of polysaccharides. This is well organized. Okay, so it's going to turn out that the slime layer is not so well organized as a capsule, but the capsule itself usually is something that you would be able to actually see. We could have done the capsular staining, but part of the reason why the capsular staining doesn't work as well is because they usually don't have a very big capsule when they're growing happily in a lab tube. Okay, and it basically just turns out that it looks a lot like a negative stain unless it has a really big capsule. Okay, but it is something that in itself is normally determined by the genetics of the cell, whether it has a capsule or doesn't have a capsule. How big that capsule is might be determined by what kind of environment it's living in. So you're going to hear this kind of information over and over, because when it comes to talking about control of bacteria, control of growth, we'll be mentioning and saying, well, remember about the glycocalyx, remember about this. When we talk about the pathogenicity of the organism, again, it's going to come up. Because do you think that having a capsule would affect the ability of an organism to cause disease, or how virulent of an organism that might be? Why? Because it's going to be a lot harder to kill it off if it has a big capsule, right? It's protected inside its capsule. If the capsule is from here to there, all the way to the end of this room, isn't it going to be a lot harder if you're trying to have an antibiotic actually get to the organism?
When you say from here to there, you mean it's that thick?
Yes. I mean, if I'm a bacterial cell, my capsule could be like the diameter all the way to the end of this room, all the way around. Can be very significant. So you see here protective advantages, resistant to phagocytosis, right? You remember what that is, right? As far as the immune system goes. Protection from desiccation. If there's a huge capsule, then that organism is going to be able to survive for a period of time. I mean, again, 14 years in a disinfectant, and we're not talking about an endospore forming organism. We're talking about Pseudomonas, like the Pseudomonas that you're working with. Of course, in some of those years, and this has been multiple times, many nosocomial infections with those organisms. Remember what that is? A hospital-acquired infection. Imagine that's where they found it, but you're using a disinfectant or an antiseptic that actually has the organism in it. I can't even tell you how many times you see antiseptics being taken off, like, I don't know whether they made it all the way to the grocery stores, but like how I've mentioned that I get those alerts from the FDA, there's numerous times that you see things that get taken off the market because they find that they're contaminated. I worked in a lab, an independent lab, where we used to test sterile products. So one of the things, like we did a lot of different things, but one of the things is as the company had their things sterilized with gamma radiation, then they would send a certain percentage to us, and then we would go through and run fluids through them and cut them up and put them in, you know, anaerobic and aerobic medias and incubate them for a week so that we could report back to that company and say, yes, in fact, the things that you had sterilized really are sterile. And we worked in a clean room, and one of the things that we would do is, you know, like I'm sure that you've seen people do this before, you use alcohol and you light whatever your tools on fire so it burns everything off on the outside. At the very end, then we would filter the alcohol, and 100% of the times organisms grew from the alcohol. So even though we were using a sterile product, we were using in a clean room, you would still get growth of organisms from the alcohol itself. I mean, we never looked to see what those organisms were because we would filter it and put it in a liquid media, so you didn't really see the culture characteristics. But like, it never didn't grow. Again, protective environment. Exclude viruses and detergents. So anything like a detergent actually usually has ingredients that help to break down things, but still, if it has a large capsule, it's going to be protected. The slime layer is usually outside of the capsule, and this is an unorganized layer. So you could have the capsule, and then on that side of that, you actually have additional layers that really start to just become part of the environment. So they're less and less and less organized until it finally gets to the point where if it's in some kind of media, it's part of that media. Okay, it doesn't have an outer edge. So you have a capsule like this, and then the slime layer, you know, could be something like that. It just has like it gets less and less organized as it goes out that way until it's kind of part of that environment. The slime layer can also help it just kind of move along in that slimy layer. And really it's kind of thought that, like with the exceptions of, because there's always exceptions, that probably most all bacteria have some kind of slime layer. So whereas a capsule is going to be only associated with certain bacteria, everything is going to end up with some kind of layer on the outside. And this shows a picture of it. Here's, you can see here that's part of the glycocalyx. And that is a capsule. Here's a capsular stain. here you can kind of see the glycocalyx going around each one of these cells. This is in the tissue. So you can see like there's some structure here. And then lastly, the S layer. The S layer is a little bit different than a capsule or a slime layer. This is a highly structured layer. It's a protein or glycoprotein. And it's kind of interesting that it's self-assembled. So it actually like starts to put itself together without even necessarily being part of the cell. That's why they're using this idea, and they're using these proteins of S layers in the field of nanotechnology, because they can find that they can use it to take medications to certain areas. I'm not exactly sure how it all works, because the person who gave us a little in service about it didn't really understand how it all worked either. And I've tried to look it up a little bit to get some more information, and it's like the information is just vague enough that I don't think that I understand it enough to explain it to you. But that whole field of nanotechnology, they've been working with this ability of these proteins to kind of assemble themselves without needing to be actually part of the cell. When you look at this gram-negative bacteria, that would be actually, outside the outer membrane. Remember, the outer membrane is that outer membrane of the cell wall. So outside of that, in gram-positives, it would be associated with the outside of the peptidoglycan layer. And also S layers are very much associated with the archaea. So this kind of shows like that structure like that. These little proteins have different shapes. This one looks like it's kind of like a hexagon. And basically, even though it's really structured like this, it still allows things to come in and go out of the cell. It's kind of a lot like that Gore-Tex fabric, Gore-Tex that they use for like rain gear and stuff like that, that doesn't let water droplets in, but if you're perspiring, it lets like that out. You guys never heard of that? Mm-hmm. Like they use it like in tents and stuff like that, and you go backpacking or camping, so it doesn't get like really like condensation inside the tent. It's like breathable but waterproof at the same time.
They do in boots too.
Pardon?
They use it in boots too.
Yes, in boots and in like rain gear and stuff like that, because it's very functional in that way. It's kind of, this is like that. It lets certain sizes in and certain sizes out. So it's very protective. And in the archaea, it's something that actually can give structural support as well. So you see again, protection, maintain shape and rigidity, especially in those cells that don't have already components that do that. So normally what gives shape and rigidity to a cell? The cell wall, and specifically the peptidoglycan layer of the cell wall. So if you have something that doesn't have a cell wall, then the S layer could be very functional for that. Also, adhesion to surfaces, protection, and then I mentioned about the potential use of nanotechnology. So that takes care of the cell envelope for the bacterial cells. How does the archaeal cell differ? An S layer may be the only component, because it turns out that the archaea don't have a peptidoglycan layer. They have something that is called pseudomurine, which I think is probably one of these. Yeah, it's kind of but some of the archaea don't have a cell wall. There's less often where you see capsules and slime layers associated with the archaea. So for some of the archaea, the S layer is really that only component. The membranes, like the cell membrane, remember how the cell membrane of a bacterial cell was that fluid mosaic model with that lipid bilayer? we have monolayers instead of a bilayer in the membrane of an archaea. So why do you need to know this? This would be something that I will be asking on the exam, something like, what are some of the major differences between a bacterial cell and an archaeal cell? So I think like last time I mentioned something, I already don't remember what it was. Do you remember what it was? Because I know I mentioned this last time, but there was something. So now there's some other things to add to that list. So you should be just making a list off to the side to make sure that you kind of have at least, probably 5 or 6 things that are really uniquely different about an archaeal cell. So different lipids, a monolayer for a plasma membrane, no peptidoglycan, usually only have an S layer. So here specifically about the cell wall, lack of peptidoglycan. So that's why an S layer would be more significant, because the peptidoglycan is the part of the cell wall that provides the majority of structure. It may have a protein sheath external to the S layer. Here is that term pseudomurine. Pseudomurine is really, remember how the peptidoglycan said in parentheses, sometimes it's called the murine layer. So that's where the pseudomurine comes from, because it's more like, this is not like a peptidoglycan layer, but it's similar. This kind of just shows some of that, how these kind of, I don't know if there's anything specifically on here that, I guess this is just showing the different types of archaea. So that's everything, almost everything outside, because we do have to talk about the fimbriae, hilli, and flagella. But now we're going to be inside of the cell. Okay, so if you're diagramming this out, you're diagramming, a plasma membrane, and then a periplasmic space, and then whatever type of cell wall it has, then a capsule and a slime layer and a possible S layer. That's where we are so far. Okay, inside then, and remember last time I said, if it lost its cell wall or part of its cell wall, it would be called a protoplast, but that term was also used to really refer to the cytoplasmic membrane or plasma membrane inward. So that's where you see that. The protoplast is the plasma membrane and everything inside. And it does have cytoplasm, just like you learned about when you learned about the eukaryotic cells. Okay, and in that cytoplasm is where we're finding this other stuff. They thought for so long, really, that there wasn't any other structural components, but they do know that there could be some cytoskeletal components in some of the bacterial cells. So, what they do might help in the cell dividing. It might keep different things in the cell in different areas. It's not really fully understood, and they definitely don't all have some kind of additional structural component. So the cytoskeleton would just be something similar to the type of cytoskeletons that you see in eukaryotic cells. This just shows some of them in fluorescent. We might have some membrane, so again, no membrane-bound organelles. However, there could be things like a mesosome, which were those invaginations or enfolding of that plasma membrane. And that could be something that could be helpful to the cell because of adding additional surface area of those membranes. So instead of having the cell membrane or plasma membrane that looks like this, you could have one that was like this. So all along those foldings give additional space for some type of physiological reactions to occur. You see these, at least as something that's known in photosynthetic bacteria that we would refer to as thylakoids, which is where you would find the photosynthesis taking place. Okay, so something like chloroplasts, but you see the little dots that are on those little enfoldings like that. So observed in bacteria with respiratory activity. It's something that, again, they may or may not have any kind of enfoldings. And remember, I said this was a little bit controversial because the only way of really understanding what's going on is by looking at it in electron microscopy and the different types of techniques that they use in order to prepare the cells to look at them also cause a little bit of change to the cells. So it's kind of gone back and forth how, much of an impact those different procedures have on what the cell actually looks like. It would make sense that they would have them, and they do know that they have some enfoldings like this. And like this, you see these in here, which are associated with those photosynthetic bacteria. So they definitely see some things like this. And who knows what other things that might do. It might be helpful for the cell in other ways as well. This next category, the inclusions, are sometimes referred to as inclusion bodies. This really, I would say, I would call everything else that might be in the cell. We still have to talk about the genetic material and ribosomes. Okay. Other than that, everything else that might be in the cell could fall into this category. If I were organizing this in the way that I would normally talk about it, this would be the last thing I would talk about. Because I would say, like, okay, well, everything else that might be in a particular cell, we're going to call inclusion bodies or cytoplasmic inclusions. Because these are usually very specific to the type of bacteria. So you see the description, granules of organic or inorganic material that are stockpiled by the cell for future use. Well, some might even be enclosed by a membrane. So again, this is another one of those things where you see, oh, there's no membrane-bound organelles or something in the cell, and yet, yes, there are things that are bound by membranes in the cells. They might have proteins, they might be lipids, they might be referred to as microcompartments. They could be glycogen, carbon. This phosphate polyphosphate is called volutin. That's the type of food storage that's associated with algae, the cyanobacteria, different types of amino acids, storage of nutrients. Here's some examples: sulfur globules, carboxysomes. These are associated with the carbon dioxide fixing bacteria. Gas vacuoles. Those are associated with some of the aquatic organisms that allow them to maintain a certain area in an aquatic environment, because some of them need to be more near the surface, some of them need to be more towards the bottom, because many of these that are photosynthetic utilize different wavelengths of light. So how that light waves come into, like, say, something like a pond environment, then those organisms will be at different layers of the pond utilizing different wavelengths of light. So when we say something about bacteria being found in every known environment, except for a sterile environment, you really can see bacteria everywhere. When you look at a pond, you will see different bacteria at every level of the pond just for that. Something like gas vacuoles then help them maintain where they need to be. Magnetic zones that are associated with some of the bacteria that have some, like, they orient themselves to magnetic fields. asking something like for you to list me, say, 4 examples or so of cytoplasmic inclusions or inclusion bodies or inclusions. So what are they? They're just everything else that are associated with specific organisms, you know, that help the functioning of that particular organism. Obviously, you're not going to find all of those cytoplasmic inclusions in every bacteria because many bacteria don't need those. Depending on what they are, they might have different types of cytoplasmic inclusions. Does that make sense? Ribosomes. You know what ribosomes do, right? Ribosomes are associated with the protein synthesis. These ribosomes are so similar to eukaryotic ribosomes, but they're different sizes. So if you look at this, if you look at the entire ribosome, there are 70S ribosomes. Eukaryotic cells have 80S ribosomes. That S, I don't know if you ever learned about this before, it's just the Stenberg unit. But it just has to do with the size of the ribosomes. They're made of smaller subunits, 16S, 23S, and 5S. But basically, what you really need to understand is just that the ribosome size is different. 70S, they're 70S ribosomes. And we could have an antibiotic or something that is destructive against these types of ribosomes that would not necessarily affect our ribosomes. So it does make it a little bit more unique. So even though they are ribosomes, there's enough difference that you could have something that affected a bacterial ribosome that did not affect our ribosomes. So the next category of antibiotics that are not as destructive to us would be the ones that are active against ribosomes. And that would be something like erythromycin and that group of antibiotics. I'm sure you guys have heard of that. I'm sure people have taken erythromycin or Z-Pax. Okay, so penicillins and that category, those work against the peptidoglycan layer. That next group, the ones that work against protein synthesis, will be not as destructive. And then, I mean, we have some other things to talk about in here too, but as far as the structure goes of the cell, the last thing is the nucleoid or the genetic material, because we have to talk about plasmids too. So the genetic material mostly consists of the nucleoid. Okay, there's going to be some other extra chromosomal genetic material that is going to be in the next step, the plasmids. But this is really usually one closed circular double-stranded DNA. Okay, so the nucleoid is an area in the cell where you have this chromosome completely supercoiled. And it's usually somewhere in the center of the cell. So if we go back, let's see. I don't know why. If we look here, here's the nucleoid. Kind of looks weird, like SpaghettiOs or something like that. There's some other pictures in there that show what it looks like in an electron micrograph. Usually somewhere in the center of the cell, you see a dense area. And they're kind of representing it here, looking like it's curled up. So if we look back on this too, you can see the ribosomes, and you put last time that there are no ribosomes outside of the capsule. So you're going to ignore that. Here's the capsule, but you can also have a slime layer on here too. So you can also, if you were diagramming it, you could diagram the slime layer. You could put something like an S layer on the outside, which would be actually underneath the capsule. But the nucleoid is just this area in the cell. And what does the area consist of? That supercoiled double-stranded DNA. Supercoiled. When you look here, here's a picture here that shows it on an electron micrograph. You can see like that dense area. That area would be the nucleus. Look at this particular picture. This is a DNA, an E. coli, in which they have broken open the cell, and here is the chromosome. They think that if you could stretch this all out, it would be about 1 millimeter. So what is the size of an E. coli? Maybe something close to a half a micrometer, right? That's how supercoiled the chromosome is. Almost like the only way that it could even fit inside that cell is to just be supercoiled, condensed like that, and really in there. If you rupture it, it's huge in comparison. Would you guys just follow along with that? So that's why supercoiling. These nucleoid proteins help keep it coiled together. But if you were to uncoil it and stretch it out, it would be so much bigger than the cell itself. Usually one closed circular double-stranded DNA molecule. Everything is always usually because there's always exceptions. So that's why when you see a cell, like a diagram of a cell, sometimes they just do like a squiggly thing in the middle like that, but other times you just see it represented as a little circle like that. Okay, like maybe it looked like that when we looked at the invasion, with the bacteriophage. And when we talk about some of the other methods in which you get genetic diversity, you'll refer to it like that because of the fact that it's a closed circle like that. I mentioned also there's other extra chromosomal DNA, plasmids. Plasmids are really important. Every semester I ask questions about plasmid on the exam, and all of a sudden not everybody, a handful of people are like, wait, I don't remember what that was. Please do not forget what a plasmid is, because plasmids are extremely important, not only just to talk about the cell, but when we talk about the transfer of genetic material, and if you were to talk about, any kind of genetic engineering or anything, plasmids are something that you probably worked with plasmids in your 107 class. Did you work with a plasmid when you did some kind of transformation experiment where I don't know whether you gave a pigment or you got an organism to become antibiotic resistant, right? In your 107 class, I know you guys did a transformation experiment, right? You guys don't remember this?
No. I didn't like it.
Did you guys all take 107 here then?
Mm-hmm.
I know you guys do the transformation experiments. Guys, that doesn't sound familiar at all? Seriously?
The onion leaf?
You're the first class that said that. Did they just not use it last semester or something? That doesn't sound familiar at all, transformation.
We need DNA.
Oh, I think but you will be hearing more about plasmids. Because plasmids are little pieces of genetic material that are found in cells. You can have multiple ones, and they're mostly responsible for antibiotic resistance. They do a lot other than that, but a lot of the antibiotic resistance that's passed from bacterium to bacterium is by way of plasmids. So it's extra chromosomal DNA found in bacteria, archaea, this is also some fungi. Usually also small closed circular DNA molecules. So you do usually see them referred to as also just a small circle, not a large circle, but just a small circle. Exit and replicate independently of the chromosome. So what's on that plasmid is like not only extra chromosomal, but we would say that it codes for non-essential properties, right? So the chromosome of the cell itself is going to code for what the bacterial cell needs, like what kinds of proteins and what it needs to survive and undergoing binary fission and all the essential qualities of the cell. The plasmid is just something that gives it additional genes to be able to do other things. One of them is to be able to survive in the presence of an antibiotic. But also you can have ones that provide genes that allow it to ferment other sugars, utilize other nutrients, have different kinds of advantages. Some of the plasmids they're referred to as conjugated plasmids because they have genes on them that code for the formation of the F pilus, or otherwise known as the sex pilus, that allows that particular plasmid to be replicated and transferred to another organism. Some of them have toxins, code for toxins, the production of toxins, so they can increase the virulence of the cell. So I most specifically will be asking you to give me some of the non-essential properties that are transferred by plasmids. And that's why I'll make a big deal about it, and then people will go like, I don't remember what a plasmid is. So it's very important, and it's something that, like, you know how I've mentioned this now a couple of times about the fact that we started off with that anaerobic, thermophilic, rod-shaped bacterium way back. We thought at one time it was the universal ancestor, and now we have bacteria living in every known environment. How did that happen without sexual reproduction? So we said already that there could be mutations and that we could have transduction. Remember what transduction was? The transfer of bacterial genetic material by means of a virus. Okay, so we know that transduction. Plasmids can be transferred from bacterium to bacterium by conjugation, by transformation. Basically, a cell could die off and leave a plasmid in the environment, and another cell can uptake that plasmid and now just has the ability to have whatever particular properties that plasmid has. So plasmids are really significant when you think again that there's only so many ways that bacterial cells can change their abilities, since there is no sexual reproduction. Plasmids are going to be something that's really essential to them. And this can happen after the fact. This is, from one mature bacteria to another. It's not like necessarily something that it has to be. Here, this particular term, which you're going to see this term again, an episome. An episome occurs when a plasmid actually incorporates itself into the chromosome of the cell. So a plasmid can come into the chromosome, it can come back out of the chromosome. When we talk about conjugation, there's going to be some different types of conjugation. Remember what happened with the lysogenic virus, where the viral genetic material went into the chromosome, and when it came out, it accidentally took a piece with it That can happen with an episome too. When an episome comes back out of the chromosome, sometimes it accidentally takes a piece of the chromosome it was attached to. So then when that plasmid is passed to another organism, it's taking essential properties of the cell as well. So it's important that you're kind of like understanding this information too, because when we refer to it, you're going to be like, wait a minute, I don't remember that. You have to remember this. Okay, so remember about all of the structures of the cell for sure, but you want to make sure that it's usable information. And by that I mean that, like, not only are you memorizing it, but you're kind of understanding it. Everybody good with that? So here, as I've been saying this now several times, genes that are non-essential. Non-essential means that they really are like giving something extra to the cell. So confer a selective advantage like drug resistance. The majority of antibiotic resistance occurs through plasmids. There are other things that the cell has that also help it, but that's definitely one of them. You could have many copies of the plasmid in the cell. You could have multiple plasmids in a cell. They found plasmids that will have resistance to 8 different antibiotics, all in just one plasmid. So if that plasmid is transferred to another cell, now that cell just picked up antibiotic resistance to 8 different medications. They're very, very significant. They're inherited through cell division as well. And then plasmids, different types of plasmids are basically classified according to usually like what kinds of genes they have in it. So if you look here, conjugated plasmids are associated with the types of plasmids that will code for the formation of the F-vellus and also then allow that plasmid to be replicated and transferred to another one. Our plasmids are associated with antibiotic resistance. Virulence plasmids have to do with toxin production. Metabolic plasmids are something that will allow it to utilize different nutrients. So this just gives you kind of some of the examples. So any questions about the things internal? We have the mostly nucleoid and ribosomes as the most important thing. Then we had some of those membranes like mesosomes, the structural components as a possibility, and then all those cytoplasmic inclusions. Now as far as like external structures, these are things that are those things that look like they're hanging on outside the cell, but they're not because they're attached actually to the inside of the cell. So the fact that they're external, I mean, they don't start from being external. They start internally. They just happen to hang off the outside. Okay, so they extend beyond the cell envelope. What is their function? Protection, attachment, horizontal gene transfer. Horizontal gene transfer is what we're talking about when we're talking about the ability of like something to occur like conjugation. So this is going to be something like the F-vellus, or sometimes known as the sex phase. And cell movement. And these basically are going to be hilli and fimbriae and flagella. And flagella, this is something that you already know something about. Hilli and fimbriae, when we look back to that cell, I don't want to go back to it, but those were all the little squiggly things that were hanging on. What's really important to note, though, is how they're attached. They're attached to the cell membrane or plasma membrane. They're short, thin, hair-like, proteinaceous appendages. The fimbriae, there might be up to 1,000 per cell, and they help attach to things. So now, again, you know, not only with the glycocalyx, but also with things that are outside, that's going to help them stick together and stick onto things. So mediate attachment. Apilus, which sometimes would be like a sex pilus or F pilus. F stands for fertility. Okay, those are like Fimbriae, except they're longer, thicker, and less numerous. So it's almost like that when you look at cilia versus flagella, something like that. The Fimbriae are shorter, more numerous. The pili are longer and fewer. And usually the main pili that you would talk about would be something that is that F pilus or fertility, or also called sex pilus, that will attach to another bacteria. And this again is going to turn out to be controversial. The most recent information says that this doesn't even happen. We just thought it happened for decades, but the idea was that a plasmid would be able to transfer between one bacterium and another. And that's what all of the photography looks like. That's what they think has happened, but it seems like what really might be happening is that it's just helping it attach, and it's just actually entering the cell another way, not really through the pilus. But if you read like the most recent literature, it says that they just don't believe it happens like this. But again, that's kind of the nature of microbiology, because when you're working with microscopic organisms, you just, like, it's very difficult to get all the information that's needed. That's why you see a lot of changes that occur. Okay, but otherwise, as of now, we know it functions in conjugation. But exactly how it functions might be a little bit misunderstood. Okay, and I mentioned with the plasmids that they had the genes that are what cause the F pilus to occur, and then also causes the replication of that plasmid so that it could be transferred to another. Conjugation basically occurs when two organisms really bump into each other. It's something that's actually really random, and then some type of chemical reaction occurs that causes a formation of this F pilus, if that particular bacteria has a plasmid that has those particular genes. Do you guys just follow that? So we'll be talking in more detail about conjugation, just like we're going to talk about transformation and transduction. But there's no way to talk about all these things without knowing a little bit about it. So this is a good picture that shows Fimbriae. So you can see them, all the little hair-like protections on the outside. They're different lengths. And then compare it to the size of the flagella. It's unfortunate that this particular slide doesn't show the pili, but if it did show the pili, it would be about this big. Okay, so see how little the Fimbriae are? The pili is usually thicker even than the flagella. So you know what a flagella is. You saw the picture of the flagella on there. The flagella is something that helps it be motile. It also helps it with attachment. It may have virulence factors associated with it. Virulence factors are just going to be different, production of enzymes or toxins or a protection that allows it to survive. If you look at bacteria flagella, they thin, rigid protein structures. You can't see them. They're small enough that even though we can look at the cell, we wouldn't be able to see a flagella. And there's a lot of detail about, I'll come back to that slide, about the structure of the flagella. So a lot is really known and understood. I'd like to show this particular picture because you can see how the flagella is integral, starting at the plasma membrane, going through the cell wall. And this gives you another opportunity to see the different